everyone. Welcome to the Around the World Advanced Therapy Medicinal Production Research in South America. Today's webinar will explore the latest breakthrough in ATMP research across South America, including innovative production techniques and regional advancements driving the field forward. I would like to remind you that all questions will be saved until the end when both speakers will answer the questions. Please submit your questions through the presentations using your QA tab that is just located to the right of the video. And also you can continue to submit the questions through the Q&A discussion at the end of the talks as well. So I want just to begin, thank you all again just to join us today. And I will introduce myself. I am Virginia Picanso. I am a researcher at the Blood Center of Ribeirão Preto, part of the University of São Paulo in Brazil. I hold a bachelor degree in biological science from the University of São Paulo and a PhD in medical science also from the University of São Paulo. My professional experience includes serving as a senior research at a biotechnology company named Veritas. I also work as a visiting research at the Drug Discovery Laboratory at Purdue University in the United States. And currently I lead research projects focused on cell therapy, including genetically modified T and NK cells. And also I'm working with the production of viral and non-viral vectors to be used in the clinical setting. Just before listening to our two speakers today, I would like to share a little bit of our experience here in Brazil, our experience manufacturing CAR T cells. We started in 2015 when we decided to design our own CAR, anti-CD19. So we designed our CAR. We established our CAR production platform, including T cells, isolation, transduction, and expansion of CAR T. And we also developed our lentiviral vector production to be used in the CAR T platform. And when we started in 2015, everything was manufactured using an open system. And during this year, we started to develop our own closed system. And in 2019, we achieved our first milestone by treating our first patient here in Brazil, in my city, in Ribeirão Preto, that is located in São Paulo state. And since then, we have treated 20 patients that are currently, and we are currently running a phase 1, phase 2 clinical trial. And along this journey, we have faced several challenges, including material importation like cell culture media, cytokines, among other materials. Also, for us was so hard to train people. We didn't have people here in Brazil that had experience to work on GMP facility. We also have so many challenges, as I told you before. We closed the system, so we are producing all the cells using an open system, and we have to close the system, and that was a huge challenge for us. And we also implemented a robust quality management and also quality control system to meet regulatory standard, and also to ensure the high quality of our CAR T cell product. And these challenges are part of the growing difficulties of ATMP development. And I'm certain that our guests today have encountered similar experience. To learn more about this topic, with a special focus on efforts in South America, we are honored to have two exceptional presenters with us today. Our first speaker is Dr. Maria Veronica Lopes. She holds a degree in biochemistry and chemistry from the National University of La Plata and completed her medical degree at University of Buenos Aires. Veronica earned her PhD from National University of La Plata and completed postdoctoral research at Leloa Institute under the mentorship of Dr. Podhaizer, as well as the University of Alabama at Birmingham in Dr. David Correal's laboratory. With a support from a CONICET fellowship, she is also a researcher at Leloir Institute, which is part of the National Scientific and Technical Research Council, the CONICET, in Argentina. So, welcome, Veronica.
Hi everyone, I am Veronica Lopez from Instituto Leloir, Buenos Aires, Argentina. Before starting my talk, I would like to thank the ASGCT for the invitation. It's an honor for me to be part of this series of talks. Our work is based on the use of oncolytic adenoviruses for the treatment of cancer. When we started our work a few years ago, we asked ourselves what the unmet needs cancer and oncolytic virus were. The tumor stroma matters. Prevent the escape of malignant cells is very important. Facilitate drug penetration into solid tumor is also important. And stimulate the immune system to help eliminate the tumor is very, very important. Avoid adverse side effects on normal organs is important too. So most unmet needs can be addressed using a tumor stroma target tumor microenvironment responsive oncolytic virus. Why the stroma should be targeted? The crosstalk between malignant cells and stromal cells support tumor growth. These cell types need to be eliminated to achieve an effective cancer cure. And there's no much treatment for stromal cells. So, with the purpose of selecting a tumor-specific promoter to drive adenovirus expression, we have selected the promoter of SPAR. SPAR protein is expressed in both malignant and stromal cells. We have analyzed several fragments of the SPAR promoter and select a 512 BP for this matter. So we construct the virus, adenovirus F512, and you can see here the result of one of the in vivo tests in melanoma tumor and pancreatic tumor. As you can see, adenovirus F512 exhibits high in vivo efficacy in both tumor types. And also, does not kill normal cells. However, we improve our version of adenovirus F512 by a new version with an insulator sequence and a new E1A mutated, which we incorporate the E1A unable to bind retinoblastoma protein. Here you can see the activity in cell lines of ovarian cancer. Okay, then we move to samples from patient of tumor of patient and incorporation of this E1A mutate into oncolytic virus shows no replication in normal tissue, as you can see in patient 13 and 14. This is normal and this is normal, and remains active in tumor samples. Okay, It's important to mention that we also observed that that virus was able to replicate in tumor samples from patients refractory to chemotherapy, like patient 15 and patient 16. Okay. After that, we added a new improvement to our virus in the spark promoter by adding 12 responsive elements of NFKP that enhanced viral replication in inflammatory conditions. Also, we add in the same promoter 3 responsive elements to oxygen environment. So we targeted with the triple hybrid promoter in combination with the fiber. This virus here, you can see the genome. This is the fiber 5, and you can see the promoter activity in melanoma and fetal fibroblasts. And the promoter activity enhance the virus activity in presence of hypoxia and TNF alpha. Also, you can see dramatic in vivo enhanced efficacy in melanoma tumor. When we change the fiber 5 by a chimera fiber 5-3, you can see the improve in vitro and in vivo in CAR negative melanoma cells. The in vivo experiment take until 80 days, so was so effective. Then we began to work with malignant cells isolate from a cystic fluid of patients, and we combined our adenoviruses with cisplatin and observed a synergistic effect, as you can see here. And then we try again with more tumor samples from patients. In this case, we use ovarian tumors, cervix tumor, and uterus tumor. Also, I test our viruses in normal uterus. This is the effect in cell line of cervix, and in most of the tumors analyzed, we observe the effect of the virus. Finally, we clone 2 immunostimulatory payloads to our virus. We choose 4-1-BBL and CD40 ligand. So this is the genome of our new virus 523. Here is the expression of the CD40 ligand, and this is the expression of 4-1-BBL. And this new virus can kill ovarian and lung and colorectal cancer cells. The new virus also was able to kill tumors in nude mice in the flank, and the eradication of tumor was 8 in 8 animals of 10. And also in a model of ovarian cancer dissemination into the peritoneal. In this experiment, we changed the CD40 ligand and 4-1-BBL by a murine 1's version, and use syngeneic Balse mice harboring murine CT26 colorectal tumors. In this case, we injected 2 tumors in the left flank and in the right flank, but only the tumor in the left flank was treated with the virus. And we observed the abscopal effect in the right flank. In this experiment, we injected the tumor in the left flank, but in the right flank we used tumor cells pre-infected with the virus, and we observed the abscopal effect in the left flank. Okay, and until here we have a virus, a new platform, which target malignant and stromal cell. The vector can sense the tumor microenvironment features such as hypoxia and inflammation. Improved system delivery achieved by replace of the fiber node. The vector was armed with 2 payloads and also can be combined with chemotherapy. At that moment, a patent was licensed by CONICET and Institut Leloir to the startup Unleash Immune Oncology. And we start to think the unmet need at that moment was the high cost of biological drugs. The use of more than one drug increased toxicity, and because of the resistance of the tumors, we need target more than one target. So we decide to address this unmet need with all-in-one oncolytic adenovirus in combination with CAR T cells. So we think that checkpoint inhibitor is important to add our adenovirus, and also a BiTE here to increase the immunotherapy. And our adenovirus would be combined with CAR T cell therapy. This is a collaboration that we have with Bonamino lab. Okay, finally, our mode is we infect the tumor, which is a cold tumor, with our adenovirus that lyse the tumor cells and stromal cells and release the antigens of the tumor. And this antigen is taken by the immune system, but also the immune system is improved by our payload from the adenovirus. Thank you very much for your attention. I open to questions.
Veronica, thank you so much for your presentation. Now let's move to our second speaker. Our second speaker is Dr. Carlos Javier. He is a pharmacist and has a PhD in biological sciences. He is a professor and director of the Institute of the Study of Inborn Errors of Metabolism at Pontificia Universidad Javeriana in Bogotá, Colombia. His research is focused on development of innovative treatments for lysosomal storage disease. Welcome, Dr. Javier.
So good morning, good afternoon, everyone. I'm Carlos Almésiga. I'm from Pontificia Universidad Javeriana at Bogotá, Colombia. I would like to thank the organizer for the invitation to be part of this webinar, in which I will share with you some of the research in the field of gene and cell therapy that is performed here in our country. So I'll first start with the research from our lab. I work at the Institute for the Study of Inborn Errors of Metabolism. It's not the whole building. Our lab is on the 10th floor of this new building of the Faculty of Science in our university. And in our lab, we work doing the research, teaching, and diagnosis of inborn errors of metabolism. And within the research line, we work on the study of the molecular and cellular basis of the disease. We also develop diagnostic methods and tools for this group of diseases. And we also work on the development of biotechnology tools for inborn errors of metabolism, thinking on the development of new treatment alternatives. So within this research line, we have mainly worked with lysosomal storage diseases, which is a group of about 70 diseases characterized by the accumulation of partially degraded substrates within the lysosome. This group of diseases of the group of disorder are produced by the deficiency of a protein or the production of a uh, mutant protein, leading to the multisystemic and progressive diseases characterized by the alteration of different tissues or affecting different tissues and producing a specific type of characteristic depending on the substrate that is accumulated within the cell. You can treat these disorders to different approaches. One of them is to restore the function of a gene or to supply a functional copy of the gene. The other one is to restore the enzyme through the use of enzyme replacement therapy, hematopoietic stem cell therapy, or molecular chaperones. And also you can reduce the synthesis of the substrate that is accumulated. So I will focus on the gene therapy strategy because it's the one that in which our lab has mainly focused most of the time. So for this, we have mainly worked on the mucopolysaccharidosis, which is a group of lysosomal storage diseases produced by the accumulation of different glycosaminoglycans. Most of our studies have been done for MPS 4A, which is a bone disease. Also, the patient presents lung disease and heart disease without central nervous system involvement. Is one of the most frequent diseases in our country. Actually, the third lysosomal storage disease in our country with about 250 patients currently diagnosed. So our gene therapy project for MPS 4A started about 20 years ago. We start with the standardization of the production of AAV vectors in our lab, and we run the first in vitro experiments of a gene therapy here in Colombia, one of the first in Latin America. And with this, we then move to improve the vector. We evaluate different promoters, trying to identify an eukaryotic promoter that help us to reduce some of the side effects that you may find with a viral promoter, and also the inclusion of an activator that increases the activity of the enzyme. We selected one promoter. We evaluated as I mentioned, the effect of the activator, that is the sulfatase modifying factor 1, also known as SOMAP1. And then we run the first MPS for a preclinical trial with this strategy. During that project, we observed that the vector is not efficiently delivered to the bone. So with the collaboration of Dr. Anane Montaño and Dr. Junji Tomatsu, we designed an AAV2 vector with a bone targeting signal, which increases the bone biodistribution of this vector. You can see here, after 2 weeks of infusion in a mouse model of the disease, most of the vector was present on bone, and this allows us to increase or significantly increase the enzyme of the activity of the enzyme in this tissue bone that is one of the most important tissues affected in MPS for patients, and also in heart, suggesting that this modification may also benefit other tissues because it's affecting the biodistribution of this vector. Then we try to increase this the efficacy of our gene therapy strategy. So we move to a lentiviral vectors. So once again, we start doing all the standardization of the production of these vectors here in our lab, and we did some in vitro experiment of this with these vectors. This was the first evaluation of a lentiviral vector for MPS. It was a study that was did by Maria Alejandra. And so we observed that with this vector, we were able to reduce the lysosomal storage in cells from patients. And we also described for the first time that there was an alteration on the autophagy in these cells that was partially correct with the use of the transduction with the vector, only with the gene, but also with the gene with the Gallan's gene plus the addition of the gene that encodes for the activator against SOMEF1. We were trying to define how to continue with our gene therapy project when CRISPR came to the scenario. So we moved from our gene therapy strategies based on the use of AAV or lentiviral vectors, and we moved to CRISPR-Cas. But we took a decision at that moment. When we started our project, the production of viral vectors in Colombia was with a GMP quality, was not like a easy task that we can perform here. So we moved to a non-viral vector strategy. In addition, that we found some research groups here in Colombia that were producing some very interesting non-viral vectors that may use as a carrier for our gene therapy strategy. So with this, we design a strategy that is based on the use of a Cas9 nickase, a plasmid encoding for a Cas9 nickase. A plasmid having the donor sequence, and this donor sequence is carrying the homologous recombination arms and a expression cassette with a promoter and the gene of interest. And we combine these two plasmids within a non-viral vector. We did the evaluation of a commercial liposome, a magnetoliposome, and a polymer. These two non-viral vectors, the magnetoliposome and the polymer, were produced and designed for research group from Colombia. So with this, we've first did an in vitro confirmation of our strategy, and using MPS for a fibroblasts, we were able to observe that after 30 days post transfection, we were able to reach 40% of the wild type levels. And noteworthy, we observed that when we use different cells or cells derived from different patients, we observe a wide variation on the results in terms of the enzyme activity and also gene expression, which was mainly associated with the mutation that was present in this fibroblasts. In this case, we observe some fibroblasts that can reach up to 80% of the wild type levels, while others only reach levels below 10. And similar results were obtained for another lysosomal storage disease, that is GM2 gangliosidosis, specifically Tay-Sachs disease and Sandhoff disease, in which we observe enzyme activity results between 2 to 10% of the wild type levels, which is significantly lower than the one that we observe for MPS4A, although we use the same gene or the same expression cassette and the same vector of almost the same strategy of transfection, showing us that we need to optimize the therapy or each strategy depending on the disease. And we also observe a wide variation between the results depending of the cell that we were using, if we use cells from different. We then did an in vivo evaluation of these strategies. First, we did an MPS for a evaluation using a mouse model of this disease in collaboration with Kenji Tomatsu. And we observed that after one administration and after 2 weeks of follow-up of these animals, we obtained levels in plasma around 30% of the wild cell levels. We were able to significantly increase the enzyme activity in different tissues, aligning the activity in heart, lung, and bone that are relevant tissues for this disease. We were able to correct and normalize the glycosaminoglycan levels in urine and bone and joint. And also we were able to maintain the structure on the tissue on the growth plate and knee joint, which was significantly altered on the untreated animal, suggesting that with this therapy we cannot only restore some of the biochemical markers, but also some of the histopathology, or maintain and conserve the structural tissues after the therapy. Ongoing projects that we have with our strategy include the in vivo evaluation of the gene therapy for stage, a project that we are doing with our partner called Turkey. And so far, the preliminary results shows an increase in the survival of the animals. We have started our project for phenylketonuria, observing a decrease on the phenylalanine levels and an increase of the tyrosine levels. And we have also started a project for MPS IIIB or Sanfilippo syndrome type B, uh, in which we have observed increased activity, the normalization of the thorax, and we have here the opportunity to evaluate a polymer, uh, which gave us the opportunity to get similar results to those observed obtained with a commercial spec. So on the other hand, I would like to share with you the results from the gene therapy and CAR T project for an institute here in Bogota, public institute here in Bogota, that is the Institute for Science, Biotechnology, and Health, ITBIC. They are working on development of a CAR T product. One of the important things here is the infrastructure that they have, that is constructed for the production of cell and gene therapies here in Bogota. They have clean rooms, they have areas to produce their vital vectors, and they have also all the quality control and quality assessment programs for the production of these kind of therapies in Bogota. And they first did the design of the vector, of the lentiviral vector, and the production or the clinical batch that they will use for the production of the CAR Ts. So they obtain titers uh, in the order to 10 to the 8 and 10 to the 9 viral particles are on the vector result for the production of these therapies. And with this, they have done the isolation of the cells and the transduction of the cells. So they isolate CD3 positive cells and transduce those cells with vectors obtaining 50% of protein expressed in the CD28 marker, and also about 61% of the cell producing interleukin 2. One of the, I think, most important results is how these cells are behaving. So these cells, they are observing a high transduction efficiency between 60 and 70% on average. These cells can be expanded, and I think one of the most important results is that these cells are showing a cytotoxicity effect. So if this is preparing all the documentation, is doing the scaling up of this process, preparing the first clinical trial in Colombia for CAR T, and actually it will be the first gene therapy trial here in Colombia. They are using a fully automated manufacturing process, and they were preparing all the dossier and all the documentation to submit the protocol to our regulatory agency that is called INVIMA within the first quarter of next year. So hopefully in the near future we will have our first gene therapy project here in Poland. Just to finish, some final thoughts about the challenge that we have in Colombia in this area. And I think that gene therapy coalition will be very important to help us to fulfill this challenge and help us to accelerate the development. So one of the first things is that we need to have a regulatory frame of gene therapy, or a specific regulatory frame for gene therapy in Colombia. We have a regulatory frame for biologic products, but we know that we need to have some specific regulations for gene therapy. And also we need to strengthen the capacity of our regulatory agency, not only to approve and to evaluate those therapies that are developed or that were developed in other countries and are approved for other regulatory agencies, but also to support local initiatives. We need to increase the training and qualification of our personnel. We have high qualified personnel here in Colombia, and I hope other Colombians that are around the world watching this webinar also are high qualified on the field of gene therapy, but we need to increase the number of scientists and regulatory personnel uh, form in gene therapy, and we also increase the number of agreement, gene transfer agreements that facilitate future developments. And finally, we need to strain and expand our local capacity for the production of gene and cell therapies, our or production of gene and cell therapy vectors here in our country. So I think that is all that I wanted to share with you. Thank you so much, and I'm happy to answer any question.
Okay, thank you so much, Carlos and Veronica, for the great
Thank you.
presentation. I think we can already start our discussion. Was so nice to see your presentation, and I think we have a very similar situation. So I'm talking here from Brazil, and probably we have similar challenges to produce those type of therapy in our countries, right? And just my first question, I see that you both use vectors, and as Carlos just mentioned at the end of his presentation, for you, how hard is to produce the vector? Thinking in a GMP setting, is it possible to you to produce? Because here in Brazil is really a problem to produce viral vectors or even non-viral vectors for use in clinical trials. How is the situation in your countries?
I think I may start. Yeah, here in Colombia, just like during the last few years, I think maybe 4 years, as I just mentioned, we have the capabilities to develop clinical grade vectors in Colombia. We only have one facility, and it's, from my point of view, is very interesting that it's a public institution with the capability to produce clinical grade viral vectors. They are working right now only on lentiviral vectors, but of course, those facilities can be used to produce other viral vectors. On the other hand, the non-viral vectors, we don't have yet the full capacity to produce clinical grade non-viral vectors, although we have a long list here of research groups working on the development, on the evaluation of those kind of vectors. So I think we have already the research capacity, but we need to expand our facilities for the production of those type of non-viral vectors. Although there are some initiatives from the government, especially here in Bogota and also in Medellin, trying to connect the industry, the universities, and the government to allow that all those three entities work together to enhance and to improve our current facilities and capacities. Yeah, but it's hard.
And Veronica, how is the situation in Argentina?
Yes, we have the same problem that you, but I guess the pandemic was very, very good because more people think about the production of virus for the vaccine, but this is the same for gene therapy. So here in Argentina, we have some few facilities to produce the virus. But it's hard to do the engineering run and to scale the virus, and this is the problem here.
Yeah, we have the same problem. So we have what we call a lentiviral facility here in the university, but it's also very small. We just have batches for the non-clinical studies and maybe for phase one clinical trials. But just for our university, for us, it's not possible to produce virus for other people because the facility is very small. And I think that it's a challenge that we need to produce more. It's just the basics of the, for example, for CAR T or for CAR NK therapy, right? To have the vectors to modify the cells. Yeah, but we still have to, we still need to invest a lot on that. And I think Brazil is doing the same thing. Here, the universities that are working with this type of therapy, they are public university, and the government is also trying to connect universities with companies, with startups to try to create an environment to develop more, right? That we don't have many things that we need. And what about the regulatory? What do you think? So here we have Anvisa, and how is in your country? How do you think that it can help you to achieve to achieve a clinical trial, to start a clinical trial, and of course to have a product at the end?
So again, I can start again. So here we have a strong regulatory agency. It's called INVIMA. We have a regulatory we have regulation for the biologic products, mainly recombinant proteins. We don't have any specific regulation for gene therapy, which difficult the entrance of this kind of products to our country, and especially the other part, the reimbursement of the therapies. Yeah, because we need to combine the approval and also the payment of those therapies, because our health systems works like any drug that is approved by our regulatory agency will be covered by the government. So that's difficult. Sometimes the entrance of this kind of product that, as you know, are like very expensive, especially for a country like in South America. So it will be very important to have a specific regulatory regulation for gene therapy products. But although we don't have that, we have already an approval of a gene therapy clinical trial for a gene therapy product. It was sad that the patient were not recruited, so the study was closed here. But it was a very milestone in Colombia, because it was the first gene therapy clinical trial that was at least approved to be run here. And that's allowed that the regulatory agency prepare for the evaluation of those dossiers, and also the researchers, we participate on the evaluation of that protocol. So that gave us also the opportunity to check how we need to be prepared. Nevertheless, we need to also to strengthen our agency so they can support our local initiatives, because currently they are not very well prepared to support these kind of initiatives that are developed from our country.
Yeah, that's right. And Veronica, do you have something to add?
Yes. We have here ANMAT. It's called ANMAT, and they are very, very prepared for gene therapy, because again, for the COVID vaccines, so they are prepared for the dossier. But we need more the facility that ANMAT is very good for us.
Okay, yeah, here in Brazil, our regulatory agents, they started to work very close to us. That was a really good effort. And because before we just submit the documents and they analyzed and they said, no, this document is not good, so you have to come back. But now we are working together with them. That is a very nice experience because we send the documentation and they say, okay, that's okay, or here you need to improve this, you need, and then you improve things or you do extra tests that they require to us. But is everything very fast, and that allows you
Yeah.
to go faster. And what they did with us, so I think this is the first clinical trial that is running in Brazil, that we produce everything in Brazil, lentiviral cells, everything we did here. And what they did, so we already we have, when we treated the four first patients, they said to us, okay, send the data, we are going to see the data, and if the data is okay, we are going to open the clinical trial. And until now is working well. We still have much to do next year, but it's a good experience. So I think they are learning, and we are
Mhm.
also learning. We didn't have many tests to release the product, and some of the tests we had to send the cells to
Abroad.
US,
Yeah.
for example, yeah, to do the test and then come back because we don't have the technology here.
Mhm.
And so most of the tests we developed in our center because it was impossible to send. And
Yeah.
then we start to develop. Now we are validating the test. We have this agreement with the regulatory agents to start the clinical trial, but at the end of the clinical trial, we need to have all the tests validated.
Oh, great.
We are working together. That's a very nice experience. And Veronica, was just curious about your research. It was so interesting for me.
Thank you.
And so solid tumors is always a challenge, right? We know that this,
For CAR
yeah,
T and for adenoviruses. Yes, I know.
for everything, because they are so hard to treat. And do you think this combination is going to work? How do you expect the combination that you mentioned, CAR-T with the AAV to target the microenvironment? How do you think that is going to improve the treatment in solid tumors?
The first thing is we planning to use low doses of CAR T and low doses of adenoviruses. So do you have less toxicity? This is the first thing that we think. And less toxicity for CAR T and less toxicity for adenoviruses. And we're planning now use intratumoral adenovirus first and systemic CAR T addition. And our adenovirus has the possibility to increase the immune system. So we hope that the bite in the adenovirus increase the CAR T activity. At least in vitro is working.
It's working. Okay, that's great. And
Yeah.
what type of target are you using on CAR-T?
EGFR.
Okay. Yeah, that's very interesting. Yeah,
Yeah.
it's a challenge to eliminate those cells. Yeah, when we think in solid tumors.
Yeah.
Let me see if we have questions for the audience. Let me. Um, no,
May I ask a question to Veronica?
Yeah.
sure.
Because I was so surprised because you are combining two strategies like viral gene therapy and cell therapy. So do you expect like you may have or like a higher risk of adverse effects since you have a virus and then a cell therapy?
No, I don't expect high
Okay.
risk because of we planning to the doses of the adenovirus use a lower doses
Okay.
that normal experiment, and the same with CAR T.
Ah, okay. Beautiful.
Okay,
Thank you.
no, you're welcome.
Yeah, that is very interesting to hear about what you are doing because it's very similar that we do right now. So we start with CAR T and now we are trying to move to NK cells because we are trying to move from autologous to allogenic, and it's even more challenge, but we are still in the beginning of this new that we call our second cell therapy. We still have to work a lot on that.
Yes.
So I think we don't have questions for the audience. Can we go to the closing, or we still have some minutes if you
No, I think is this kind of opportunities is very important because we can share what we are doing in the region, but also I think is the opportunity to unite efforts. So because
sure.
I do not agree that each country will need to have the facilities to produce every type of vector. I think we can think like for instance, Brazil can produce lentiviral, Argentina can produce adenoviral vectors, we can produce adenosuicidal, associated vectors. So I think it's important to organize those kind of things because we are a huge region, but if every country needs to start from zero each project, it will cost a lot and it will take a lot of time. And I think we need to unite efforts to accelerate the development of these therapies in our country, especially because of the patients. That we need that the patients benefit a lot of those kind of therapies. And for me, it's very good to hear all the experience from Brazil about the approval and about how they lead with the local developments and how they enter all the therapies that have been developed in other countries. I think is a great example. How can you like combine the experience from the local researchers with the developments that have been done in other countries?
Yeah,
I
for sure.
totally agree.
Yeah, and when we think that those type of therapies are very very expensive, especially for us, right? That we live in a poor country, and it's so hard for the population to have the access to this type of therapy, and it's important to us to develop our own uh, product. And of course, we need to join forces and learn one with the other, right? To try to develop. That is. Let me see if I see some. Oh yeah.
I guess that Virginia froze.
Well, as Virginia, yeah.
Yes.
Okay, I think we need to improvise. Okay. Veronica, do you have like approved products in Argentina for gene therapy?
Not now. We don't have approved any product now.
Oh, okay.
We sent to approve in USA, but we are waiting for.
Oh,
Yeah, but yes, it's so hard because the research is okay, but then you have to move to another thing, and you need more help for that.
oh, okay, great. And and what about the startup? Because we also open on a startup, but for other therapies for recombinant proteins. But what about the startup that you start with the development?
Oh, my
What?
connection.
Sorry?
Oh, No, yeah. We were improvising, Virginia.
Okay. Virginia?
Yeah, because she already came back. No.
Virginia? You
Yes.
listen?
Virginia, yeah.
Yes. Okay. No, that's problem. So your question was
about the startup, because you mentioned on the startup.
No, startup, no.
No. Oh, okay.
No, sorry.
Okay. Just I understood that the patent was licensed to a startup.
Yes. We license, but I'm not work in the startup.
Okay.
Yes.
Okay. Got it. I understand.
I'm so sorry, my connection was off.
Okay.
So someone sent us a question asking about if we have a product, if this product is going also to be approved by FDA, for example. If I can talk about Anvisa, I guess yes, because Anvisa, she's following FDA. So everything that we do, we also have international certifications in our cell facilities. So of course we don't have a product yet, but I believe that once we have, it's going also to be also available for the other countries. And what do you think about?
Yeah, here too, because most of our regulatory process is based on the FDA and EMA regulatory process, especially at the FDA, and all of our industry, pharma industry here, and all the facilities follow the ICH protocols, and also they receive certifications from international entities. So I think that if we can produce a product for gene therapy developed in Colombia, it will be for sure, or at least we can be confident that it can be approval from other international agencies.
Okay, let me check if we have more. I think no, that was the last question that we have.
We have here a question for Masato Yamamoto. Maybe you, Carlos, can answer the question, because you have a GMP product.
Can you share a question? I cannot see it.
Yes, if you GMP products are available for FDA application in the USA.
As I mentioned before, I think we can. It will be. It can get approval from FDA or EMA. Yeah.
Okay, I think we can go to our closing. So I would like to thank you again. Thank you so much for this great discussion. And I would like to thank everyone that was listening to us, and thank you again for sharing your expertise and for the great discussion. This event was recorded, and you receive an email with a link to watch the recording when it's available. And also, I like to announce that the previous ASGCT Around the World series webinars are now available to watch on YouTube. Please feel free to view the videos and click in the links on the Extras tab. Finally, ASGCT values your feedback and welcomes suggestions for the future sections. Please take a moment to complete the survey, link it on the right of the screen under evaluation. Thank you so much.